My name is Bambi de la Rosa. I'm the Healthcare Artificial Intelligence Principal Investigator within Micron's Advanced Computing Solutions Group. And I'm honored to engage in such meaningful collaborations and am humbled to invite to the stage the next set of panelists who are truly on the front lines of innovation and pushing forward progress in the space of precision medicine. So let's give a warm welcome to Dr. Joe Gray, Dr. Manish Kohli, and Dr. Hamid Ratej. Have a seat.
Thank you.
So I think to get going, the best thing to do is to get to know each of you and the work that you do. So maybe we'll start with you, Joe.
So my background is in engineering and nuclear physics. I was convinced as a youth that that's what my calling was. But as I proceeded on in my education, I began to encounter the fact that while we knew a great deal about the area that I was working in, as my friends and my parents began to develop diseases like cancer, I became appalled at how little we knew about those diseases. And I was given an opportunity early in my career to basically switch and begin to take some of the tools from the area of physics and begin to bring those to bear on helping us to understand how it is that disease arises, cancer in particular. And it was sort of the beginning of thinking about precision medicine. How can we use that information to do a better job of treating? And so as time went on, I began a career of developing measurement tools that allowed us to understand deeper and deeper what's going on in cancer. I'm a fan of the Lord Kelvin quote, "If you can't measure it, you can't fix it." And so I've made a career out of trying to do a better job of fixing it. I grew up during the era of genomics, spent 20 years or so working in that area. But about a decade ago, I came to the conclusion that just knowing about the components of the cancer were not enough, that we were really going to have to understand what it is that the DNA codes for and how is it that those proteins are organized into molecular machines, into organelles, into cells, into tissues, into people, in a way that helps us to understand what the emergent characteristics are of those collections of proteins that are either normal or abnormal, and to help us to understand how it is that we could correct or fight some of the abnormalities that arose. And so that took me up to Oregon Health and Science University at my Cancer Institute, and what we've done up there is to put together really a very broad suite of measurement tools, nucleic acids, proteins, but all the way up to the multi-scale architecture of tumors using electron microscopy, light microscopy, anatomic imaging, and so on. And the idea is to bring all of those tools to bear on the individual cancer and to help us understand how it evolves, how we're going to treat it in the instant, and how we're going to evolve treatment as the cancer evolves to escape what we're trying to do.
Amazing, interesting work. Manish?
Well, I'm delighted to be here. Thank you for inviting me. This is a fundamentally different landscape. As a medical oncologist, my training was in the pre-genomic era, so to speak. In 2001, as you know, the genetic code got cracked, and what happened before that was very piecemeal, slow. We used to practice a lot of empiric clinical sciences, which we continue to do many times, even now. But what's happened in those last 19 years is that as we have fundamentally learned more and more about the central dogma of life, which was alluded to just now, DNA, RNA, and proteins, there has been a mushrooming and a therapeutic abundance that has come about to be. We have, for example, in breast cancer, 21, metastatic breast cancer, 21 FDA-approved drugs. In kidney cancer, metastatic kidney cancer again, 17 FDA-approved drugs, and so on and so forth. And our job now has become how to pick any of these drugs for the individual patient in front of us because none of them are 100%, and match it to the central dogma of the cancer cell of the patient in front of us. And that's the precision medicine that got me very, very interested in trying to be curious about, trying to do something about, because I've been humbled as a medical oncologist over and over and over again in these 20 years when we play a cat and mouse game with the cancer, put it into remission, it comes back again, like you showed in the video for the patient over there, and then we try something new, put it into remission, it comes back again. But we want to do that more smartly, so we don't give the toxicity of our drugs, but give the good effects of our drugs. So that's where I've come to be. I've trained for the last 25 years in that. I was at Mayo Clinic before joining Moffitt Cancer Center this year, and I'm the director of the Personalized Medicine Institute over there now.
Awesome, thanks. Having
First of all, thank you so much for inviting me to this panel. I actually come from a completely different background from Joe and Manish. By training, I have been an RFIC designer. In fact, in my past life, I did a lot of the work that Micron does, semiconductor-based design, whether it's on the memory side, whether it's on the wireless communication, optical links communication. And I was lucky enough that about 7 or so years ago, that this opportunity at the company that I am with right now, GenapSys, to be able to go and apply the exact same science and engineering and a semiconductor and apply that to the healthcare business. And not just any healthcare, to apply that to DNA sequencing. And that's what we have been doing at GenapSys. We have developed a new way of reading DNAs. For those of the audience who may not be as intimately knowledgeable with DNA as I wasn't about 7 years ago, I didn't know what DNA stands for. So I know there were 4 letters, but what that I had no idea. But said, okay, we know binaries, zeros and ones in the field of electrical engineering. Going to A, B, and C, and D, what is the difference? There's no difference, right? We could just A, B, C, G, T, doesn't matter. They're all. But we got there, and I learned that we need to start reading at the molecular level. Every DNA base, every nucleic acid is about 3 angstrom, 0.3 nanometer. In fact, the DNA is the most condensed and energy-efficient storage medium. The human DNA is made of about 3 gigabases, and when the way that it coils, it will be a few nanometer across, and it's smaller than any geometry that today the semiconductor, as advanced as it is, and the industry that I come from, has been able to achieve. So being able to go and read every 3 angstroms of incorporation of a DNA and be able to say that's what the DNA construct is, it was mind-boggling for me to start, and still is. But I think the good thing is that the engineering field has been very successful to break through many barriers, to be able to give the tools that the scientists and the oncologists like Joe and Manish need to be able to help the cancer patients that we just saw a movie of them. And it's an honor to be part of that industry.
So do you know what DNA stands for now?
Today, yes.
Okay, making sure we're making progress.
But don't ask me to say it.
Okay, I won't. Maybe later. So maybe we should start with Manish.
Yeah.
What is precision medicine?
So very conceptually speaking, right drug for the right patient, for the right cancer type, at the right time, so that there is a right outcome. That's precision medicine. Mm-hmm. In trying to get through all of those rights, there are a number of hoops that we have to go through. And that's what we are trying to unravel for the individual patient in front of us. And how can we do that? It takes team science for doing this. You have to have the clinician who basically are the front sort of face with the patient. But they need the tools from the engineers, from the chemical scientists, from the physical sciences, and all of them, and the bioinformatic people, the coding structure of the DNA and what it reveals for that particular patient, all putting together. And then they probably need something of an instrument for not just detection, but for guiding that next treatment. So that's precision medicine in a nutshell. A few years back, these engineers, a couple of engineers called Robert Hooke and Anton von Leeuwenhoek, put two glasses of lenses together, and they made something called a microscope. And suddenly the clinician said, oh, we always suspected of something called a cell, but here, now we can see it. I think it's the engineers who are again going to help the clinicians in trying to get that paradigm shift in getting to more precise medicine. And so it really is a team effort in this foray as far as I'm concerned.
So we often hear, sometimes interchangeably, precision medicine versus personalized medicine. So maybe, Joe, you can help us disentangle what is the difference, if there is a difference.
So my definition of personalized medicine is basically taking the diagnostics and the drugs or the diagnostics that predict a particular drug and applying that to the individual patient. Mm-hmm. Now, typically, when you start that process, you'll get a solution. But unfortunately, in advanced cancers, in many cases, the tumors will evolve over time to escape the control. Even if you were effective in the first place, they may escape control. Mm-hmm. And so the idea of personalization is, what do we do now? What has gone wrong with the tumor? What are the new therapeutic vulnerabilities? How can we adapt the next drug to be effective against that? And so our whole point of personalization is we are going to treat the patient forever. And we will evolve the treatment based on our diagnostics to control it as long as we can.
So one ingredient of any successful AI endeavor is data. We've been hearing about data all day today for good reasons. And in the CERN, we were hearing about 30 to 40 million collisions a second. So I wanted to kind of get a sense of what is the type of data in the precision medicine space. So not only just size, but also a little bit of context around, what kind of data are you using in the precision medicine space, or do you intend to use in its full complexity? So maybe I'll start with you, Joe.
So what we try to do is take on the discovery mode of measuring as much as we can about each individual cancer. And so we certainly look at the DNA, we look at the RNA into which it's transcribed, the proteins into which it's translated, and we then use a variety of imaging tools to help us understand what the organization of the proteins are into cells, into tissues, into organs, and so on. And so that takes us into the realm of electron microscopy, super resolution light microscopy, conventional microscopy, CT imaging, PET imaging, MRI. And so for the individual patient, at one time, for one treatment point, we'll probably measure 10 to 20 terabytes of data.
Wow. And are you able to use all of that data at the moment?
Well, so that, of course, is the challenge. The first issue is how can we get it all organized and linked to the clinical outcome information that we also have to be acquiring. And then we have to be able to search for the signatures within that mass of data that are clues about how to make it actionable, that is to say, to identify drugs that we think will be effective for that patient. And so that's a huge computational challenge, and I would say that we are very much in the infancy of being able to do that. Right now, far too big a data set, and it takes far too long to analyze it. And so that's one of the reasons we're excited about the Micron collaboration.
So I know you gave a little example of how technology has kind of pushed forward the space of precision medicine, or better made us able to kind of get these very detailed measurements. So I kind of want to talk maybe to you, Hamid, to discuss like what was some of the motivation of GenapSys, and kind of what's the purpose of your technology and software, particularly in the space of precision medicine.
I actually tell you, the way that I joined GenapSys, this is when GenapSys was just incubated outside of the Stanford University. And when I was speaking to our founder CEO, convincing me that that's the right place to go, and this was a story that he told me then, this is about 8 years ago, that, and you guys should probably correct me if there's any truth in it, that if there's a cancer patient at a time, you come in, God forbid, you have a cancer, they go, the best drug that we have is drug number 1, we apply to it, it has a 30% chance that it would cure you. And God forbid it doesn't, then it may several months pass, the second drug doesn't work, third drug, and every time that, obviously every one of these drugs have so much side effects and the fact that you're losing time to fight against cancer. And that's where the concepts of precision medicine started for me 8 years ago, that if you could provide the information to the scientists, to the health industry, that they could recognize that for this specific cancer, that's the specific gene that has been affected or has been mutated, and now this is the drug that you have to use. If the third drug that on average has maybe 5% chance of cure, but that a specific case would have had 90% of a cure. Just because it statistically previously was assumed that it's 5%, they did not use it. So that was the starting reason behind GenapSys to start. And then if you look at it throughout the history, obviously we have gone, in fact, DNA sequencing has surpassed Moore's law, the way that the semiconductor has done it. DNA sequencing has done it way faster than the Moore's law. And the NGS, next-gen sequencing platforms that are available today, and GenapSys is one of the earliest of the technologies that has come out with a solution. The intent is to be able to put the DNA sequencings closer to the point of service. If every doctor's place can have a sequencing platform, if every emergency room can have it, if every hospital can have it, if every research labs could have it. To give you an idea, today there are more than 400,000 molecular research labs in the world, and less than 1% of them have DNA sequencing technology in them. I was chatting with Joe before coming to here, and he was saying that they need to use service providers for their, and it takes several weeks before they send the samples to them and they get it back. If you could have a technology at your fingerprint that is cost-effective, it's easy to use, and it can generate the data that you're interested in, then you could get the information faster, and then you could cure the patient faster. And those are the underlying reasons behind what GenapSys started to do, and then what we have delivered and created over the past 7+ years. And of course, the next component, as Joe mentioned, is that you have the data, what do you do with it? So the way that GenapSys has approached is that we create the data, we make it available in real time on the cloud. And that's where you enable it for any type of machine learning technologies that can run in the background on your compute servers in the cloud to run through the data and sift through it and give you the answer you're looking for. And the next step, of course, is make that artificial intelligence that comes up with data, make the data available to the greater audience and make that more powerful. And that's our mission.
That's where you can use deep learning accelerators. So I want to say, Micron's been a leader in the technology industry for over 40 years, and that doesn't happen by existing in a silo. It's really enabled by deep engagement with the broader ecosystem. So I thought maybe we could have you talk a little bit about the collaboration, about the smart clinical trial, and then leading in to kind of give us a little context into the video we just saw about the collaboration.
Right, so the main focus of the collaboration right now is helping us to try to parameterize, if you will, the three-dimensional data that we're generating on individual cells using electron microscopy. And one of the things that one appreciates when you go into the world of electron microscopy is that you have a very data-intensive, feature-rich world, and we've been struggling over the last few years to understand and be able to visualize and to be able to quantify the individual organelles that exist in a cell. Many of these things actually are diagnostically informative if you can actually quantify them. Right now, we mostly do it by hand. That's clearly not scalable, and one of the things that we do know...
How long does it take to do by hand?
Well, we've acquired one image 2 years ago, and we're still working on it. So there's more information than humans can dredge out of that in a lifetime, in that single image. And so, obviously, that's not the way that it's going to be done. What we have to do is to begin to apply tools like some of those that are being developed in the world of AI, and to learn what it is that humans are doing while they're doing this kind of annotation, then break the problem up into multiple pieces and to automate it. If we can't accomplish that, we really just can't use the data. But I think that we're very excited about the collaboration, because it's taking us down the road of being able to automate what is right now an impossibly time-consuming and, quite frankly, subjective task.
Well, and what I really like about the example of the collaboration is that, in AI we hear a lot of hype, and so sometimes it's hard to differentiate, like separate hype from reality. And in cases like this, it's a narrow application of the sense of AI. It's not general intelligence, but it's fulfilling a very meaningful role, and it's a synergy within your existing pipeline. So it's ways that we can kind of optimize tools and existing things we have now to actually, help accelerate insight into your workflow. But it's not it's a synergy. It's the doctors, clinicians, and everybody coming together to work on the problem. So I think that's really beautiful. So I want to we're cutting it close on time. So I want to end with, which I know it goes by fast. We could talk all day, I think, in this topic. So I want to end high level, though, on what is each one of your vision on maybe the role of AI in precision medicine, but more kind of this marriage of technology and health in a real tangible way. What do you view? What's your vision of the future of this?
So I think that we have the technical ability to generate vast amounts of informative data today. And buried within those data sets are things that can be discovered by AI that humans just can't see. And so I think that we're moving down the road of being able to perhaps not understand, but at least identify actionable signatures in disease automatically and quickly in a way that will, quite frankly, revolutionize how well we're able to treat complex diseases like cancer.
Save lives.
Save lives.
What I'd like to do is to get your AI machine learning platform, look at the Library of Medicine or any other data source that's already over there. Make you guys give me a target of a particular advanced cancer patient who has many drug options in front of him or her, and give me that signal. Then go to Hamid, get a blood sample from this patient, sequence that particular target, go to a chemical or an electrical engineer, make a device like this, which would be able to get that signature in a drop of blood 5 years or 10 years from now, find if that target is there, and then treat the patient according to precision medicine based on electrical gadgetry of this kind, or the sequencing that came from there, which was identified by the AI learning target, and treat the patient more precisely in the future. Is this doable? I think the rapid pace of technology in different fields is going so fast in parallel, although I think in 10 years or so it will be doable. So that's how I'd like to practice.
Yeah. I mean...
They already said what I should do. No, actually, it's exactly what we intend to do, and that's exactly what we are doing. And this, to achieve what we discussed, it's exactly the path that has to be walked through. And our goal is to be able to create a data that can be consumed with the artificial intelligence. a requirement for AI to function is data. If absence of no data, the AI cannot do anything. And the goal is for us to democratize DNA sequencing to the point that there is enough data to help us both discover and cure different diseases. To just give you another statistics, today less than 1 million people, or around 1 million people perhaps, has been their entire genome sequenced. If you look at it in the US, that would account for a fraction of a percent, 0.3%. And if you look at it in the world as a whole, you're talking about 0.015% of the people have been. And that means that we have not even collected enough data. We are a few orders of magnitude away from the size of the data we have to collect before the AI can then help us to solve through it. And I think that it's an open field. There's a lot of opportunity for creativity, for innovations, and the path that Micron is taking, I think, is right in that direction.
Well, as we've said before, it takes a village. So we're honored to walk along with each of you in this journey, and let's talk afterward about that idea.